Fc region of the antibody is the bit that determines how long the antibody lives. You can actually engineer it. We know the amino acids that drive that longevity because it's binding to a receptor that recycles it. And we can influence that, increase or decrease the half-life of antibodies. We can use the antibody specifically to target to a cell, and you can join a small molecule onto it so you get delivery. You can also take toxic molecules and stick them on an antibody that's expressed in tumours and use that to target toxins to tumours. That's called, there are different names for it, but it's called targeted cell killing, cytotoxic cell killing. Should we go?
Oh, go next. Okay. Can you tell them, when you were talking about EPO, could you tell them, do you know generally how much money that's made for Amgen?
Oh, it makes a lot of money for Amgen. That's one of our biggest, that was our first ever product. And EPO, when we cloned EPO, that was back somewhere between 1980 and 1983, which actually, if you go ahead, this one here. So this is a schematic of really how biotechnology got going. So way back when, Watson and Crick deciphered the genetic, the structure of DNA. After that, some really important experiments had to be done once you got DNA to understand the sequence and how the sequence translates through to protein. Did you guys learn about that? Did you learn about overexpressing artificial proteins and how they used it to crack the code?
No, we did not talk about that.
So it's a really nifty experiment when they did it the first time. They knew it was, the genetic code AGTC, but they didn't know how that translated to the proteins they got out at the other end. So somebody went to this laborious work of isolating all the components you need to make protein, so it's cell-free protein production, sticking it in a tube, adding a DNA template, which they artificially made to be all A's in one sequence. And they found when they got all A's, they got a protein made of all one sequence. Okay? So then they went back and changed one and saw what they get. So they worked out that codes work in threes and what each of those codes read out as when they did it. So that was the cracking of the genetic code. The next thing that came after that is we needed to be able to clone genes. We needed to be able to identify a gene that we were interested in and express it in another system to make a protein be produced. And that happened in the late 70s and at the beginning of the 80s. And we were one of the first company, there was us and another company who first expressed a recombinant protein and showed that it had activity. And that was erythropoietin. So we've been working on erythropoietin since then. It was a long time after that, maybe 10 years after that, something like that, when that first went on the market. And it went on the market incredibly quickly because the FDA was excited about being able to do this and being able to treat anemia, because anemia killed people. Anemia doesn't kill people very often now, but it did back then, particularly patients with kidney disease, because they have very crippled EPO production. And when hemoglobin gets down to a certain point, you literally can't breathe and you die. So the FDA was very interested in helping get that to patients, so it got accelerated approval. It makes, I'm just going to guess and say between $2 and $5 billion a year right now.
So it's made up for its research cost?
Yes. Yes, it's made up for its research cost. And research was much cheaper in those days because the path to regulatory approval was shorter, because the more drugs that go through regulatory approval, the more cautionary tales come out of it, the more regulatory guidelines get added and the longer things take, which is a good thing for patients. When you're making a drug, it means it costs a lot of money. It takes, about $1 billion to make a drug, generally. It takes people like me about, 7 years in the lab, I'd say. And then it needs to get into development and get made. The hepcidin project that I talked about, the basic research took 8 years. When we first started to do...
Oh yeah, I just asking how many medicines in the market have you worked on yourself?
Well, I've been at Amgen 10 years and it takes 10 years to make a medicine. So, I've worked on, I arrived at the company right when RNS was being made. I worked a tiny, tiny bit on a new version of GCSF that's on the market right now. I worked on a drug called Nplate that's hitting the market right now that makes platelets, so helps patients who can't clot. But I would say that a lot of people in biotechnology if one of the things you work on makes it into the clinic in your career, you've done well. So those things I just told you about, they were things that were already pretty close to a success when I started working on them. If you take something from discovery and take it all the way through and get it in people, you can be very happy with yourself. We normally say we need something like, with small molecule projects, it's somewhere between 10 and 70 projects, and me and a whole team of people work on one project. When it's a discovery project, maybe it's only 2 people. When it gets a little bit further, when you start to understand that it may actually target the disease, because when you know, all you know is you've got a protein that has an activity, and you think, I wonder if it will do this in this disease. So you build all kinds of in vitro models and models in mice to address that. And it's got to go all the way through the mouse models, and then it's got to go all the way through monkeys usually, before it gets anywhere close to people. And, you know, if you had one project, that would be fabulous. If I ever discover something that makes it into people, you know, and it treats disease, I couldn't expect more than one of those.
That's what I was going to say. As a researcher, to have one that goes all the way to market, that would be a career, right?
Yeah. And I really would say that most people at Amgen, you're okay if none of your projects ever make it, because you know that it's just a case of trying enough projects to get one that makes it. So, for instance, if you want to intervene in a particular pathway, like in the iron metabolism I was talking about, you might have to target 3 or 4 points on that axis or more to get the point that works. Because if you work on one protein and you intervene, and it actually happens, there's a feedback loop that nobody knows about. Doesn't matter what you do to that. It was this protein down here you needed to intervene with. Maybe this one's harder. So, I mean, that's why it takes absolutely years of trying to understand all the proteins in the pathway to understand where it is you even need to target.
Okay, Althea has a question, but we would have, if you want to be open for any more questions, we have like about 10 minutes. Okay. And just to give you a time before they have to be off on their next adventure. Okay.
Yeah, often, so the level I'm at now, I have a group that, I have several groups under me, and they all work on different projects. And you might have somebody who has 2 or 3 projects because they're very early stage and you're not quite sure what's going to work. So maybe you've got 3 projects and you work on them and you see which one picks up and starts going, and then maybe you sideline the other 2 and work on it. So I would say somebody who first starts working at Amgen, maybe, you know, fresh out of a PhD program, they'd have 2 projects. But they might help a couple of people out on other things too.
For a job like yours or an opening job, is it like, I don't know, like a 9 to 5 kind of job, or is it like, what are I'm trying to picture how the job's like. How long are you actually in a lab for like a session?
That depends on the person. Some people work day and night, you know. I know people of my level who work day and night. I personally have kids. I don't work day and night. I try and do about 9 hours in a day, and then maybe I'll check in on a few things at night, maybe check in on a few things at the weekend. If I have a project deadline coming up, if I have a big presentation coming up where I'm trying to get resources to do something else maybe, I might have to work more. So some people work 9 to 5, some people work long hours. And I think success is more about how you prioritize than the number of hours you work.
So when you guys are going to start a new project, do you just pick a disease and try to find a drug to fix it, or is it like more not as general as that?
It can be. So for instance, Amgen can decide we want to target this disease. We'll send people to a few meetings to learn as much about it as we can. They'll spend months reading the literature and trying to come up with what they think is a unique hook on that disease. Or it can be as simple as, I would say part of my job is every week to look through the scientific literature and see what's out there that just, you read something and go, but I think it applies to this disease over here and it doesn't look like these guys have thought about it yet. So sometimes it's the sort of off-the-wall thinking you get. And what I would say is the more sort of risky something is, the harder you have to try and persuade your management that you should be able to do it. But the risky stuff is usually the stuff that really nobody else is doing. So I would say that as a company,